Hi guys, David here with another science update. So today we're going to talk about some new research in the field of bacterial engineering and how this may influence the way that we approach dealing with environmental plastic pollution. In particular, we're going to talk about a common plastic called PET, or polyethylene terephthalate, and this is one that you run into a lot because it's one of the most common plastics in single-use food packaging and packaging for medicine and things along those lines. So the article says, since its first synthesis back in 1941, PET has gradually emerged as the world's favorite food-safe plastic due to its robustness, chemical inertness, and durability. Although regarded as non-toxic and 100% recyclable, single-use convenience-sized PET bottles have made PET plastic the third most collected debris in beach cleanups in more than 100 countries. It's very interesting that it says it's 100% recyclable, but it says that, for instance, the PET recycling rate was estimated to be only about 30% in 2015 in the US. So that's a pretty interesting topic in and of itself. There have been a lot of interesting exposés on kind of the gaslighting that you could say the plastics industry engages in, kind of the public relations efforts and all of their messaging surrounding the idea that, oh, we're very environmentally friendly, we're committed to recycling. But the reality is that, you know, this very, very common plastic was only being recycled at a 30% rate as recently as 2015. And it came out in 1941, so the industry's had over 50 years to kind of figure out how to recycle this and how to encourage this in the populace. But the reality is it's just not happening. So the idea that plastic is anything other than environmentally unfriendly is just, it's just amazing that people are still messaging that. And it's interesting, it says that in 2016 around 485 billion PET bottles were manufactured and around 583 billion are forecast to be produced in 2021. And so we're anticipating that people are going to be making more and more PET, and it looks like if that recycling rate stays low, like in the 20s or 30s, then this is going to be an increasing problem in the future. It's not going away. The plastics problem is actually increasing. So this paper spends some time looking at the chemical structure of PET, polyethylene terephthalate. And you can see here, this website shows basically this is the chemical structure of PET. And you can see that there are brackets surrounding it, and that means that that's a repeat unit. So plastics are formed from polymers, which are basically long strings of repeating chemical structures. And the composition of PET is two groups, this terephthalate group here and this ethylene group. We won't go too deep into it, but talking about the structure is important because there are two main approaches to recycling PET. One way is to recycle PET just like you would imagine recycling any other plastic, which is you can kind of do things like shred it and then reform it and make new things out of it. But then the other way is the bacterial approach, where they actually will use bacteria to basically eat and digest the PET down to its constituent parts. And this chemical approach basically involves the use of enzymes to cleave the bonds between the red groups here and the blue groups, which sort of breaks it down into these small molecules, terephthalic acid, which we can see here, and then ethylene glycol, which is shown at this bottom structure here. And so basically, once you have those chemical structures, the idea is that you could put together a group of bacteria which would eat these small molecules and break them down into harmless materials. That's the goal, but it's a lot easier said than done. This type of research got a big boost in 2016 when they discovered a new bacterial strain that could secrete two enzymes, PET-ACE and then MHET-ACE, which enable the microbe to grow on certain types of PET as its major carbon source under some conditions. So then the idea is, and what the focus of this particular research on is on enzymes in another bacteria called Pseudomonas putida, which is basically able, under some circumstances, to actually use the ethylene glycol and the terephthalic acid as fuel and break it down completely into harmless products. So this particular paper took a deep dive in looking at the particular protein TphC, which is responsible for binding and then chemically converting the terephthalic acid, which is a critical component of making this, basically, this molecular breakdown of PET happen. And what you can see here is what's called a ribbon representation of an enzyme. And an enzyme is a protein. And so what a protein is, you'll recall, is basically a string of amino acids, which are small molecules, and that string of amino acids can basically bend and fold in different ways. And this particular drawing is basically showing a certain way to visualize what this looks like. And you can see things like these green kind of flat or pleated ribbons, and then you have these red ribbons, which are sort of spiraled. Those are beta sheets and alpha helices, and those type of structures basically can fold together in different ways to form this globular structure, which is the actual structure of the enzyme. What's really interesting is that when they characterized this, they found that there were basically two halves of the structure, and there was this area in between the two halves, domain 1 and domain 2, which they're calling the substrate binding pocket. And this is actually where the terephthalate actually fits in and is bound by the enzyme. And then while it's there, you can see that certain parts of the enzyme come into contact or very close proximity to the terephthalate, and that's where the chemical reaction happens. And I thought this was a really great figure because it kind of spells that out. So what they did in this paper is they were actually able for the first time to crystallize this particular enzyme and characterize its structure to a greater degree than has been done before. And they also engaged in a number of studies sort of on the genomic development, so the connection between this enzyme's structure and how it was coded in the genome, to try to get a better understanding of how we could improve the functionality of this enzyme. Because the idea is if you can improve the functionality of this enzyme, then you could presumably engineer a bacteria that would more efficiently break down the terephthalate. So I thought this paper was interesting because even though it's looking at basically one small building block of the overall puzzle of breaking down PET naturally, it does suggest that if we can improve the functionality of these enzymes individually and perhaps engineer certain bacterial strains which have more of these enzymes, then you could imagine a situation where in the future we might be able to actually engineer a bacteria which could be cultivated in landfills to break down PET. So far, it looks like the sort of bacterial breakdown of PET has to be carried out under very specific conditions, non-native conditions, it says in the paper. So in other words, the first problem or the first thing that you have to do is you have to get a bunch of PET together in a specific location with specific resources at hand to deal with it, which is actually the same problem that we have when it comes to just normal recycling of PET, right? I think the problem with recycling of PET is not that there's no way to do it. The problem is that it just ends up getting thrown away instead. And so I don't think that this sort of approach as it is, even if we improved it a little bit, is really going to change the world when it comes to getting rid of excess PET or preventing environmental pollution, unless in the future, maybe 10, 20 years, we could get our bacterial engineering to such a level that we could actually engineer a bacteria that could survive in a landfill condition or out in nature and actually completely live off PET and break it down entirely. If that was the case, then I think we would basically solve the plastic pollution problem, or at least, maybe in landfills. There is a thread of irony even in that. The thing is about PET and other plastics is that one of their best properties, one of the things that make it so good, is that it doesn't break down easily, right? So, and that makes it like a good food packaging material, for instance. So if you actually did do a really good job of engineering a future bacterium that could break it down easily and you release that into the environment, you basically would have created a situation where you were undermining what made the plastic material so good in the first place. And then you might need to invent a new plastic for food packaging that doesn't break down. And so that is kind of a funny thing. And I do think that it points to the fact that there's probably a limit to which technology is going to solve the problem of, for instance, ocean plastic or environmental plastic pollution generally. I think we need social change, and we might even need policy changes in order to combat this at the end of the day, because as I've said, there's really pretty major downsides to almost all of the technological solutions. Well, anyway, I thought this was a thought-provoking paper. If you enjoyed the video, I would appreciate if you would like and subscribe, and I hope you'll join me again in the future soon. Bye.